Hello, my name is Kiwon Park, and I want to introduce quantum dot through delivery system. Do you know QLED? QLED is a product explicitly developed by Samsung, one of the world's top companies, and it displays that use quantum dot. Like this, quantum dot is a substance that is attracting attention in the display market. They are semiconductor particles and few nanometers in size, and they have optical and electronic properties from the larger particles due to the quantum mechanics. Quantum dot has many unique characteristics. When the particle size is nanoscale, it will cause quantum confinement effect, macroscopic quantum tunneling effect, and size effect. Furthermore, quantum dot has optical properties. Let's take a deep look at each feature. First one is quantum confinement effect. In nanoscale, it has discontinuous energy levels by particle spaceforce. When energy comes, electrons go up to excited state. This is followed by the process of relaxation, during which photons can relax non-radiatively into a lower line state or recombine and re-radiate. They become luminous. Next is quantum tunneling effect. In classical mechanics, surmounting a potential barrier requires energy which is higher than barrier. However, in quantum mechanics, wave function can propagate through a potential barrier which has higher energy than total energy of particle. So probability of particle is non-zero and out of potential world. Last one is size effect. As we can know from energy of particle in the box, energy is h squared n squared over 8ml squared. Because of L factor, when size decreases, energy can increase. So small size quantum dot leaves shorter wavelength right. In other words, it gives off a very precise color depending on the size. Nowadays, many anti-tumor drugs in clinical tests have various side effects, such as non-selective toxicity, poor targeting. Due to the lack of any bioluminescence, tumors can be difficult to be located and removed, particularly on small size scale. Therefore, it is highly desirable to create a means by which cancer cells could be targeted with high specificity and simultaneously imaged in vivo. To avoid this problem, drug delivery system is acceptable because physical and chemical property, physical response, and biological characteristics of drugs are changed due to the role of the carrier. Quantum dot is one of the candidates to resolve this problem. As a new kind of inorganic nanofluorescence probe, quantum dot has showed outstanding advances in the long time multicolor fluorescence imaging and detection. The development of quantum dot labeling promotes the research in the nanodrugs in cellular, even at live animal level. The development of fluorescence imaging technology of quantum dot and the therapy-based multifunctional nanodrug is expected to apply to diagnosis and treatment of cancer. This is an example. The work of some researchers focused on fabrication of polyacrylic acid-coated Cu2OH PO4 quantum dots that exhibit a strong NIR photoabsorptive ability as a 3-in-1 multi-purpose cancer teratonic system. A combination phototherapy of photodynamic therapy PDT and photothermal therapy PTT along with photoacoustic imaging modality was achieved. Under NIR excitation, a local hyperthermia and reactive oxygen species were generated utilizing the synergenic PTT-PDT therapeutic effect. Also, these quantum dots presented themselves as good candidates for the in vivo photoacoustic imaging on the tumor site for realizing an imaging-guided therapeutic process. 23 iodine-doped carbon dots, monodipole spherical nanoparticle over 2.7 nanometer in diameter, were synchronized by some researchers in order to be used as compute tomography contrast agent and fluorescence probe. This iDope quantum dot displayed wavelength-dependent excitation and stable photoluminescence, superior X-ray attenuation properties in vitro, and good biocapability. Quantum dot can be used for studying the drug release. Before we see the example, we have to know some concept PrET. PrET is pulsar resonance energy transport. It is mechanism describing energy transfer between two light-sensitive molecules. A donor molecule initially in its electronic excited state may transfer energy to an acceptor molecules through non-radiative dipole-dipole coupling. The efficiency of this energy transfer is inversely proportional to the 60 power of the distance between the donor and acceptor, making PRED extremely sensitive to small changes in distance. Traditional methods of studying nanodrug release in viable mainly determine the total content of drugs by HPLC-MS, which means leakage chromatography. However, these methods are often problematic at actual work. For example, it is difficult to quantify free drugs and encapsulated drugs separately, and there are technical difficulties in quantifying some trace drug under the background of complex organisms. Essentially, the quantum dot PRED method provides a new way to solve this problem. The principle of quantum dot PRED method is when the fluorescence emission spectrum of quantum dot and the excitation spectrum of fluorescence receptor overlap, and the spectral distance between the donor and acceptor is close enough. The quantum dot state of excitation can be used as the fluorescence donor to transfer fluorescence energy to the acceptor, which results in fluorescence quenching of the quantum dot donors and fluorescence enhancement of the acceptor. This energy transfer usually occurs when the distance of intermolecules is 1 to 10 nanometer. The efficiency of the energy transfer closely relates to the distance between each other, namely, the smaller the distance, the higher PRED efficiency. This technology has been successfully applied in the interaction studies of biological macromolecules, immunoassay, nanosensor design, and other fields. Recently, the researchers have made some progress at nanodrug release. Thank you for listening.